everyone, and welcome to today's Keynote webinar titled The Impact of Clinical Practice Gaps on the Implementation of Personalized Medicine in Advanced Non-Small Cell Lung Cancer. This webinar is a part of the 11th annual event in the Genetics Week virtual event series. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located on the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar, or from the lobby within the event. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the Abstract tab from the menu at the left of your screen and follow the process to obtain your credits. Now let's get started. I now would like to present today's speaker, Daryl Pritchard, Senior Vice President of Science Policy at Personalized Medicine Coalition, PMC. For a complete biography on our speaker, please click on the presenter window that's located directly below the media player. Daryl, you may now begin your presentation.
Welcome to this presentation on the impact of clinical practice gaps on the implementation of personalized medicine in cancer care, part of the Genetics Week virtual event series 2023, hosted by LabRoots. This presentation is part of the Precision Medicine track. I will be discussing the value of personalized medicine approach in cancer care delivery, the challenges to implementing biomarker testing-based strategies, and I will be sharing the results of a study measuring the impact of clinical practice gaps on the delivery of precision oncology in advanced non-small cell lung cancer. Thanks to our host, LabRoots, especially Melanie Chan and Lynn Brainerd for coordinating the virtual event. And thanks to all of you, our audience, who share our interest in this topic and in a better understanding and appropriately advancing personalized medicine. I am Daryl Pritchard, Senior Vice President of Science Policy at the Personalized Medicine Coalition. The Personalized Medicine Coalition is an education, advocacy, and evidence development organization based in Washington, D.C. We represent over 240 members of all different stakeholder types: the biopharmaceutical industry, the diagnostics industry, laboratories, patient advocacy organizations, clinical providers, both academic health centers and community providers, think tanks and thought leaders in the personalized medicine field, information technology organizations, and data banks, as well as payers. All of these stakeholders come together with a shared mission to advance personalized medicine. My presentation today stems from a series of Personalized Medicine Coalition initiatives to better understand and address challenges associated with the integration of personalized medicine into healthcare, including a key study to examine the impact of various clinical practice gaps associated with diagnostic testing-informed personalized medicine strategies on the treatment of lung cancer. Before I present the results of the study, I will provide a broad background of the issue, which we will discuss today. First, a requisite introduction into any discussion about personalized medicine and genomics. What is personalized medicine and how does it improve healthcare? So I have on this slide a definition of personalized medicine that was developed by the Personalized Medicine Coalition. And it focuses on the use of molecular diagnostic tests to determine which medical treatments will work best for a patient, for an individual patient. Now, you may have heard multiple terms used to describe personalized medicine, often referred to as precision medicine or stratified medicine or targeted treatment. There are different nuances to some of these terminologies, but we will use them interchangeably today in our conversation. Basically, a precision oncology approach, when we focus on cancer care, includes the use of these molecular diagnostic tests to determine what is the best treatment for a particular patient. But by combining the data from these tests with an individual medical history, circumstances, and values, healthcare providers can develop targeted treatment and prevention plans for that patient. The precision oncology value proposition is clear. What's supposed to unfold here is that if we implement a precision oncology approach, getting a right treatment to a particular patient early in their care, we should have improved clinical outcomes. And from these improved clinical outcomes, we should lower systemic costs. So this is improvements to both the patient at the patient level and to the system, where we avoid downstream progression of disease and costs associated. Now, I'm going to bring up a case here on genomic testing in oncology, and this, I hope, will set the stage for the conversation about clinical practice gaps. So despite an expanding body of evidence supporting the clinical utility of genomic testing, it really does remain underutilized in clinical oncology practice today. And when utilized, genomic testing has associated implementation challenges that can disrupt that value proposition, that can mitigate its value a little bit. I'll point to a study that was published in 2019 by a group at the Fred Hutchinson Cancer Research Center in collaboration with the Personalized Medicine Coalition, Latte Stoughton et al., utilizing a database of over 191 U.S. clinical care delivery organizations and data in lung cancer from 2011 to 2016 that showed that using genomic testing instead of standard of care single gene testing was associated with an increase of identified actionable mutations from 23% to 30%. Now, since 2017, since this data was collected, there have been the development of a number of more diagnostic markers that have increased the amount of lung cancer patients that have actionable mutations to about 70%. Still, only about 8 to 10% of US non-small cell lung cancer patients receive genomic testing, despite 70% of patients potentially having an actionable mutation that would drive targeted therapy. So that increase in identification of actionable mutations can lead to more targeted therapy use. However, we see here, and I'll see if my action works, we see here that despite identifying about 30% of patients with actionable mutations, still only about 21% of patients receive targeted treatment. This is a clear example of a clinical practice gap. When we discuss the value of genomic sequencing compared to single gene testing, we look at cost effectiveness and overall outcomes. And you can see here that the cost of doing genomic sequencing is slightly higher than single marker genetic testing, which is the standard of care. And there's a slight but insignificant, a small but statistically insignificant increase in expected life years gained for patients that do this. Now, I will mention that this is at a population level. So any individual patient may have had a very significant improvement in outcomes. However, at a population level, it's a small increase in life expectancy. The incremental cost effectiveness ratio of a genomic testing approach rather than single gene testing is about $148,000 per life year gained. Now, for those of you who don't know what ICERs are, incremental cost effectiveness ratios, basically a threshold is established at about $150,000 per life year gained for the additional cost of a healthcare technology to say that it is cost effective. So you'll see here from this study where we looked at real-world data in 191 different healthcare delivery institutions, we got a borderline cost-effectiveness ratio and a small but insignificant increase in life year expectancy or improved outcomes. Hardly the result that we would expect to show the value of using genomic testing. It was a bit of a surprise to us when we turned up these results, because by and large you would expect genomic testing to have a greater value for patients. However, what was really pointed out here, and what I pointed out earlier when we showed that a lot of those patients with actionable mutations weren't actually getting the targeted treatment, is the presence of a clinical practice gap. The population level assessment of cost effectiveness was about threshold level, barely considered cost effective. But about 30% of patients with actionable mutations, for which more effective targeted treatments are available, still did not receive those targeted treatments. Since this was a model using real-world data, we were able to determine what would those ratios be, what would those outcomes look like if all of the patients who were eligible for a targeted treatment received it. In this case, that cost effectiveness ratio would drop to about 110,000 per life year gained, which is widely considered cost effective. And the outcomes, that measure of life, of additional years of life, would become measurable and statistically significant. Now, this only includes patients that received actionable biomarker test results but failed to get the appropriate treatment. Many other patients don't even receive biomarker test results due to a whole nother set of challenges. But a study just published this year in 2023, using a Spanish reference center looking at tracking a population of lung cancer patients to look at the cost effectiveness of genomic testing, found that the cost effectiveness now in 2023 could be as high as 27.5 thousand per life year gain, which is, with no better way to say it, a slam dunk. It's a very cost-effective approach for the delivery of care in lung cancer. However, I want to point out that in this Spanish study, the patients were assured, they were tracked and assured to have the delivery of precision medicine. It was a clinical trial. It was not in the real world. It wasn't practice-based using real-world data. So that practice gap still exists. The reason I point this out is because, showing data from 2017, we saw that we could do a lot better, that using a genomic testing approach could be very effective, improve outcomes, and be cost effective. Now, using data for a tracked population, we see that it's absolutely clear, a slam dunk in providing value, both to the patient and to the system. However, these challenges still exist, and that's what I'm going to focus on. Although the science behind development of precision oncology medicines has clearly shown that that better treatments are available, it's still significantly impacted because of an ill-prepared diagnostic and clinical ecosystem. Navigating the complex precision oncology pathway is fraught with inefficiencies in implementation that lead us to a failure to unlock the full potential of precision medicine. Some of these I just pointed out. Many patients who have actionable mutations do not receive the appropriate targeted therapy. Others don't ever get biomarker testing in the first place. Top of that, the average delay for adoption of a new diagnostic test that measures biomarkers that could guide treatment decisions is about 4.5 years. This is in lung cancer. This is highly variable between different cancers. Lung cancer is a cancer where there is a widely established precision oncology approach. So things could be even worse in other cancer types for the delivery of precision oncology. This complex clinical care pathway with multiple steps from diagnosis to treatment is leading us not to be able to fully realize the value of these improved scientific developed treatments. So at the Personalized Medicine Coalition, we postulated that a better understanding of the extent of the impact associated with this clinical practice gap on patient care can help us inform strategies to develop more consistent precision oncology approaches. So basically, we know we have great medicines. Now we need to invest in making sure we understand how to use them appropriately. So the Personalized Medicine Coalition worked with partners at Diaceutics, a diagnostic information and development company that has an incredible data source, which I'll explain, to look at the impact of different steps of the clinical practice gaps at different steps along the precision oncology delivery pathway. We published a report last year, at the end of last year, end of 2022, in JCO Precision Oncology. The objective of the study was to examine the practice gaps associated with biomarker testing-informed personalized medicine strategies in non-small cell lung cancer. Again, we targeted non-small cell lung cancer because there is an established precision oncology pathway, and felt that this was a good example of where we can identify some of the challenges in implementing the biomarker testing strategy that leads to targeted therapeutic use. The data source I mentioned was from Diaceutics. It's called the Diaceutics Diagnostics Therapeutic Data Repository, a multi-source database that consists of commercial and Medicare claims, as well as laboratory data. And by marrying these two sets of data, including real-time lab data that's been de-identified at the patient level, we were able to look at a coverage of 340 million lives across the United States. Within the claims portion of that repository, a population of over 38,000 non-small cell lung cancer patients that were newly diagnosed and actively managed in 2019 were identified for practice gaps analysis. So I don't want to bury the lead. The top line findings here is, despite the lengthy history of targeted treatment availability in non-small cell lung cancer, the study found that 644 out of every 1,000 newly diagnosed lung cancer patients, 64.4%, did not receive a personalized treatment. They did not make it through the precision oncology pathway. And the reason why I present this as 644 out of 1,000 is because there's a linear progression of drop-off here that makes me have to change the denominator in determining percentage points of patients lost at every step. So the easiest way to say this is, out of every 1,000 patients, 644 were lost. In this case, overall, 64.4% of patients. That means only 35.6% of lung cancer patients make it through a precision oncology pathway. Knowing that targeted treatment is a much more effective and safer treatment pathway, this is unacceptable for our system. Less than 40% of patients actually getting the best treatment that's available to them. Now, among the patients who were potentially eligible to receive a targeted therapy, almost 50% are lost to factors associated with biomarker testing. Among those that did get biomarker testing and received results, nearly 30% were not prescribed the appropriate targeted therapy, as I mentioned before. And that followed through with this data set, as well as the data set along with the data set from 2011, 2016. That has not improved much over the last few years. So we looked at the loss of eligible patients to the delivery of personalized care at 7 clinical steps along the precision oncology care pathway, starting with biopsy referral, how many patients had gotten a biopsy in the first place, challenges around biospecimen collection. The third step was challenges around the evaluation or pathology of the biospecimen. The fourth was failure to order a test. The fifth clinical practice gap was the challenges with the biomarker testing performance. Something went wrong with that test. The sixth practice gap was challenges around reporting, usually the turnaround time associated with the lab results. And finally, failure to apply the appropriate treatment based on results of the test. So we looked at how many patients were dropped out of the precision oncology pathway at each of these 7 steps. Here's the methodology of the data approach. We started, like I mentioned, with the Diaceutics data repository, including over 500,000 patients in 2019. And from them qualified over 38,000 newly diagnosed lung cancer patients in the United States in the year 2019 that were actively managed. This really captures a majority of all of the newly diagnosed lung cancer patients that year in the United States. From there, we looked at each of the practice gaps where patients were lost along the pathway, and using this data source and a data source from real-time lab data. And each of the practice gaps, we saw how many patients were lost to attrition along the clinical oncology pathway. So I'll walk through the results of this, the impact of each of these practice gaps one by one. So the loss of eligible patients to the delivery of personalized care at step 1, this is the biopsy referral. About 6.6% of all patients, so 66 out of 1,000, never received a biopsy. Now, for this particular practice gap, there's not too much that we can do. Many of these patients were diagnosed very late in their care, and it was deemed by their physician or their family that they didn't even want to get a biopsy because they were too advanced and that they believed that it would not do any good. But there are a portion of these patients that were at high risk of having a biopsy collected because of the procedure to do a tissue biopsy that may have benefited from a liquid biopsy. So there are some of these patients that, if we adhered to a precision oncology approach, should have been able to advance further along the precision oncology path. But here we're losing 66 out of 1,000 patients. At step 2, biospecimen collection, there were problems around collecting the tissue. It was either insufficient tissue supply of an initial biopsy or a rebiopsy, and in some cases a rebiopsy wasn't able to be performed, or there was an insufficient amount of tumor in the tissue biopsy collected. Here, another 131 patients out of the 934 that had gotten the original biopsy were lost to the precision oncology pathway. These are pre-analytic processing challenges. At step 3, another pre-analytic processing challenge about tumor load overestimation. So the biospecimen evaluation. How many patients were dropped out of the precision oncology pathway because the pathologist misjudged the biopsy and it was unusable for biomarker testing. We lost 14 out of the 803 patients that originally were advanced to this stage. At the next step, biomarker test ordering, we lost a lot of patients. So this practice gap involves physicians not ordering testing or initiating treatment before testing was ordered or the test results were received. This happens frequently, and at this stage, we lost 142 more patients out of the 789 that had advanced to this stage in the first place. So nearly 18 or a little more than 18% of all of the patients never got a test ordered. At the next step, for those that did get a test ordered, the 647 patients out of the original 1,000 that did get a test ordered, another 116 were lost because of the biomarker testing performance. Either the quality was not sufficient, the test was not performed due to some processing challenges, or the test's performance sensitivity was inaccurate and it led to a false negative test. Here again, we lost 116 patients out of the 647 that advanced to this step. At the sixth step, test result reporting, we examined how many patients were lost due to turnaround time problems. This is usually associated with genomic testing, and other types of biomarker testing don't usually have as severe a problem here. But another 21 patients of the 531 that had advanced to this stage were lost because of turnaround time problems. If it takes more than 2 weeks to get a test result, we assumed that treatment was initiated before the test result was returned. And again, 4% of our patient population had a problem with the test result reporting. In the last step, and I'll go into more detail in this step because we discussed it up front and it's consistent here, we found that about 30% of patients who did get a result for their biomarker test still did not receive the appropriate targeted treatment. And this comes for various reasons, and I'll discuss that on the next slide. But here are patients that were able to have their samples collected, have tests ordered, the appropriate biomarker tests ordered, get through the processing, and deliver results that were appropriate and correct about the patient that still didn't receive a treatment based on those results. And here we lost nearly 30% more of the patients. A little more detail on this. What are the reasons why a patient would not receive a targeted therapy despite an actionable test result? Along the right side of this figure, you see some of the issues that continuously are talked about and reported. Here we looked at a survey to see what were the primary reasons why a physician or an oncologist would not order the appropriate targeted treatment based on those actionable test results. And those include reporting issues. Many oncologists are unwilling to prescribe off-label. So if there was a biomarker that in lung cancer that was telling about a targeted treatment for a different types of cancer, they did not use it, so a lack of FDA-approved indications. For some doctors, there was a lack of awareness of the targeted treatment option and or guidance, primarily through clinical decision support tools that did not include the appropriate targeted treatment. The decision analysis tool. Also cited were social determinants of health and access disparities. In some areas, these tests or the targeted therapies aren't available or aren't considered for populations because there's a lack of access. Therapy cost, insurance coverage concerns led some doctors to not prescribe those treatments, and different comorbidities or contraindications could also lead to this, to a lack of patients getting the appropriate treatment. Here we found about 14.3% of those patients with biomarker test results that indicated the use of a tyrosine kinase inhibitor targeted treatment failed to get that treatment, and 11.6% of patients with PD-L1 tests that indicated the appropriate use of immunotherapies did not receive that immunotherapy. Incorrect test results led to about 3.3% of patients not getting the right treatment. So overall, the message is that we really need to focus on the clinical delivery of targeted treatments and the precision oncology approach, biomarker testing. Here, overall, you can see out of 1,000 patients, only 356 made it all the way through a precision oncology approach. where we lost 6.6% at the first practice gap, 14.5% of patients at the second practice gap, nearly 2% of more of the patients at the third practice gaps. We lost over 18% of our patients due to lack of ordering the appropriate biomarker testing. We lost another 18% of patients because the biomarker test performance was problematic. And these are all issues which can be worked out, and we need to invest in working these challenges out so that we get better use of biomarker-led treatment decisions. We lost another 4% due to turnaround time, and 30% of patients that got actionable results still did not receive the targeted treatment. So what are the conclusions here? This was a very important study and very well read and accepted by the personalized medicine community. It's a wake-up call. It's making very clear that we have invested a great deal in the development of new targeted drugs in oncology, especially in lung cancer. And these drugs are more effective and safer and improved outcomes for lung cancer patients across the board. As many as 70% of lung cancer patients have actionable mutations that can lead to a targeted therapy. And that targeted therapy is going to likely improve their outcomes over standard chemotherapy and radiation therapy. This is a wake-up call saying we need to learn how, as a system, to deliver these products appropriately and optimally. The study provides healthcare system administrators, policymakers, and the oncology community with needed data to understand and target steps in the precision oncology pathway process where patients are losing the chance for these targeted treatments. The insights from this study, we hope, can inform efforts to optimize biomarker testing in clinical practice and therefore help drive the delivery of personalized medicine to all cancer patients, not just lung cancer patients. This data is applicable across different cancer types. Potential areas for focus and strategies for reducing clinical practice gaps and really addressing some of these issues include at the pre-analytic process stage, at the biomarker test ordering stage, and in the targeted treatment decision-making stage. First, we should talk more about, as a community, and work towards developing best practices to ensure tumor sampling, handling, and testing is efficient. We also need to improve practice integration and cross-stakeholder communication, including laboratories, as a key function. We need to make sure that samples are handed off appropriately and that all of the community, all of the healthcare workforce that is moving a patient through a precision oncology approach seamlessly handles their information and processes samples and results and information appropriately. This includes ensuring clear and timely reporting of test results. This is an issue that should not be too difficult to really improve if we can bring the community together to recognize the problem and do something about it. This also includes improving clinician awareness of testing and interpretation of results, including improved clinical decision support that is seamlessly included in an electronic health record so that the test results are automated and clear to the prescribing physician. And this also includes addressing coverage, reimbursement, and affordability challenges for tests and treatment to make sure that access is for everybody and not just for those who can afford it. So with that, I will end the conversation or end the presentation and hopefully begin the conversation. I'm happy to take any questions or comments regarding the practice gaps and the study.
Thank you, Daryl, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. All right, let's get started. Our first question asks, what strategies can we employ to address clinical practice gaps around pre-analytic sample processing and to ensure biomarker testing is optimized?
Well, thank you, Kaylee, and thanks to the audience for participating in this session and for asking all of the questions. I'm very thrilled to be here and to speak with you today and answer any questions that you might have about this unique challenge in the delivery of personalized medicine. So what strategies can we employ to address clinical practice gaps around pre-analytical sample processing? I think this is the area where we can make the most progress the most rapidly. First and foremost, there are a set of pre-analytic processing guidelines put out by the College of American Pathologists called the CAP checklist. And we must assure that we comply, that our laboratories and our oncologists comply with recognized tissue handling pre-analytic processing guidelines in this CAP checklist. That is something that all labs know they need to do, but we need to have quality assurance to make sure that they're being done. It's also important to involve pathologists at each of the stages in collection and handing off samples to the appropriate laboratory personnel. Many of the challenges that we have come from problems in simply handing off a biospecimen to the next person along the chain. Finally, I'll say that it's important to ensure that reimbursement for tumor content assessment services and tissue handling occurs so that we don't have a financial squeeze or that there are efforts to save money that might impede or mitigate good sample processing. Again, Kaylee, I think we can make sure that we do these pre-analytic processing challenges on the front end, and that will go a long way to making sure that these samples are moved forward and that biomarker testing occurs optimally.
Wonderful, thank you. I have another question for you here. This question asks and states 30% of patients with actionable biomarkers not getting the appropriate TRX is way too many. How can we optimize and better deploy clinical decision support?
Thank you, Kaylee, and thank you for the question. This is an issue around clinical decision support. It's interesting, and this is a problem that I have just shown has existed for years. How can we improve clinical decision support? One of the key first steps that I think we need to do is find a better way to integrate biomarker test results into electronic health records. This is not normally typically done. In some elite systems, they do have biomarker test results integrated directly into EHRs, but for the most part, there are challenges around IT processing that preclude this. On top of that, clinical decision support systems can be implemented at each healthcare delivery institution that can remain updated, that know what are the current biomarker tests that are relevant and make sure that those results follow through in the chain of custody from biomarker test ordering to notification or reporting to the treating physician, and then to appropriate treatment prescription. So in this case, with an implemented clinical decision support system that assures that biomarker tests are reported, there could be a flag or warning if a prescription then that is added to that electronic health record doesn't comply with the testing results.
Sorry about that. My microphone was muted there, so you probably didn't hear me. Apologies. This question asks, what practice gaps should we invest in addressing first, and will helping more easily address practice gaps help to address downstream challenges? So sorry about that.
Thank you, Kaylee. And I wanted to indicate to you that I couldn't hear you, but now we've got it. So we're back on target. This is an interesting question about what we should invest in addressing first. There's a lot of clinical practice gaps here, and there's a lot of challenges. It could overwhelm the system if we think, oh, we need to handle all these things at the same time. Therefore, there may be a strategy to ensure and invest in some of the lower hanging fruit and some of the challenges that where we can make an impact in improving optimal care strategies that it will help downstream, just as the person who asked the question suggested. I would say that some of these challenges upstream around pre-analytic processing and most importantly around biomarker test ordering are things that we can really make an impact in improving right away. And if we do improve those practice gaps, if we do assure optimal processing and sample handling, as well as automated or reflexive biomarker test ordering, then some of these other challenges downstream will be easier to handle. So I would go forward to say we need to assure that we handle our samples and process them appropriately through standards in care and standards in processing through the CAP checklist and others, including pathologists, so that there's smooth handoffs of the sample. That is a more attainable first step where we can make progress and impact the delivery of care rapidly. Then I would say most importantly is that test ordering. If we could have reflexive or automated test ordering associated with biospecimens. So when your biospecimen is collected for lung cancer in particular, there are genomic testing or a series of single gene tests that include all of the biomarker testing that is necessary to make a treatment decision are included. That's automated. That will go a long way to make sure that we don't lose all these patients along the pathway. Oh, you're muted again.
Goodness gracious. So sorry about that. What patients are doing the best in successfully navigating the precision oncology pathway? And are there significant disparities? And what policies and practices can help assure equitable delivery of appropriate high-value personalized medicines?
So thank you, Kaylee, for that question as well. Excellent question. I think this is on a lot of people's minds. Disparities in healthcare definitely occur in precision oncology. The disparities that we saw within this project included that most of the patients that even received biomarker testing were, as you would expect, non-Hispanic white. Only about 9% of patients who received biomarker testing in lung cancer were Black, and only about 1.5% of the patients who received biomarker testing were Hispanic. This is a clear underrepresentation of those communities achieving the same standards of care as non-Hispanic whites. This is probably not due to any racial or ethnic factor. It's probably due to the hospital systems and the communities for which they receive care. And we need to assure that the delivery of care, that we are handling these strategies to better implement personalized medicine in those community healthcare delivery systems that are seeing these underrepresented populations and delivering their care to them. We can't, the feeling and the sentiment is that personalized medicine isn't truly personalized unless it's personalized to all patients, not just some who have access to it. And in this case, we want to make sure that we address these disparities in care, and the processes that I talk about that strategies to better implement precision medicine need to be applied both in the academic health centers and elite healthcare delivery institutions, as well as in the community centers that provide care to all different socioeconomic groups.
Great. Well, thank you so much, Daryl. It looks like that's all the time that we have for today, so we're going to go ahead and wrap up here. Daryl, do you have any final comments for our audience?
My only final thought, Kaylee, is that we have invested a great deal of research and development and financial resources into the development of targeted therapies that work better for cancer patients. Now it's time to make sure that those therapies are being used most appropriately and optimally. If we're going to make the investment on the front end, we need to make sure that to follow through and make that investment on the back end so that these things are delivered appropriately and that every patient gets the best care available to them.
Absolutely. Well, Daryl, I want to thank you one final time for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until May 18th, 2025. Labyrinths will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.